Hi, my name is Vincent Hong, and the disease I researched is called Listeriosis, more commonly referred to as Listeria. Listeria monocytogenes was first described by E.G.D. Murray in 1924. He described them based off of 6 cases of sudden death in young rabbits and published a description with his colleagues in 1926. In this description, he referred to the organism as Bacterium monocytogenes. However, Harvey-Perret changed the genus name to Listeria in 1940. Listeriosis was not recognized as a significant cause of neonatal infection, sepsis, and meningitis until 1952 due to an outbreak of listeriosis in Halifax, Nova Scotia, involving 41 cases and 18 deaths, mostly in pregnant women and neonates, from the consumption of coleslaw that had been contaminated with sheep manure. Mortality rates of this disease: if listeric meningitis occurs, mortality may reach up to 70%. If septicemia, 50%. From perinatal or neonatal infections, greater than 80%. Even during the early administration of antibiotics, 20 to 30% of patients infected will still die. The most notable case is bluebell. Out of the 10 patients, 1 in Arizona, 5 in Kansas, 1 in Oklahoma, and 3 in Texas. All were hospitalized and 3 were found dead from their single-serve product called Scoops, chocolate chip country cookie sandwich, and the Great Divide bar ice cream. Shortly after the first few cases, production was stopped voluntarily by the bluebell manufacturers in Oklahoma, South Carolina, and Texas. Ironically, most infected were in hospitals for other reasons and consumed the infected ice creams during their stay and then expressed listeriosis. On May 14th, 2015, Bluebell Creameries made verbal agreements, and in these agreements they said that they were going to do rigorous facility cleaning, sanitizing, revised testing protocols, revised production policies. Unfortunately, the verbal agreements seem to not hold. There was a recent recall of products produced in their Alabama plant that had potential for contamination. This is the taxonomy of Listeria. The genus Listeria currently has 6 species, and out of the 6 species, only Listeria monocytogenes is consistently associated with a human illness, specifically Cerovars 1/2a, 1/2b, and 4b. This is the infectious agent. It is important to note that the organism does not synthesize flagella at body temperatures. Common detection methods is the Anton test. The Anton test consists of inoculating a culture on the conjunctival sac of a rabbit or guinea pig, which causes serious keratoconjunctivitis within 24 hours. Keratoconjunctivitis is whenever the cornea is inflamed, typically due to dryness. Listeria species can go on auger containing sheep blood because of the characteristic small zone of hemolysis that can be observed around and under the colonies. The FDA method of identification and detection was revised in September 1990 and requires typically 24 to 48 hours of enrichment. It takes 5 to 7 days to identify, but they're looking into recombinant DNA technology that can potentially require only 2 to 3 days. Currently, the FDA is collaborating in adapting its methodology to quantitate very low numbers of organisms in food. Clinical testing can be diagnosed by blood, spinal fluid, or amniotic fluid that looks for the bacteria. What is Listeria? Listeria is a foodborne disease, and once the bacteria has invaded the body, Listeria can travel through the bloodstream, but most of the time they're intracellular acting, therefore they're found in cells. 5 days to 3 weeks after ingestion, Listeria can access to all parts of the body, including the central nervous system, heart, eyes, and other locations. Transmission. An example of transmission is consuming unpasteurized milk or contaminated milk, which is found in most cases. However, other methods are possible, like consummation of food that has been in contact with fecal matter of animals, contaminated soil, or contaminated water without being washed or prepared properly. The disease can be transferred to the fetus during pregnancy because Listeria can be found in the placenta and utero and directly transferred to the newborn at the time of birth. Typically only infects humans that are pregnant, newborns, and older adults. There are 4 distinct clinical syndromes. Listeria can proliferate asymptomatically in the vagina and uterus, usually symptomatic during the 3rd trimester. Symptoms of this include fever and headaches. More severe symptoms can lead to miscarriage, stillbirth, and preterm labor. The second is neonatal infection. Neonatal infection can be divided into 2 groups: early onset sepsis and late onset meningitis. Early onset sepsis is acquired through the utero and can result in premature birth, and late onset meningitis is acquired through vaginal transmission. CNS infection. Listeria favors the brain parenchyma, especially the brainstem and meninges. It can cause nerve palsies, encephalitis, and meningitis. Seizures is also a common symptom. The most common is gastroenteritis, which can produce foodborne diarrheal disease, which is non-invasive. Patients present signs of fever, muscle aches, diarrhea, nausea, headache, stiff neck, confusion, loss of balance, and convulsions. And this is the most common whenever the bacteria contaminates an immunocompetent person. The mechanism of infection. It is first helpful to note that Listeria monocytogenes belongs to a group of bacteria that can penetrate into and survive and reproduce inside host cells, and not only the professional phagocytes but virtually any nucleated cell of the body. The bacteria is a foodborne bacteria, therefore it must be able to survive in the stomach fluids and absorbed into the body by invading the intestinal epithelium. Once the bacteria has invaded the host, the bacterium produces hemolysin and phospholipases, and can leave the phagocytic vacuole and enter the cytoplasm. Due to the unusually long incubation time, the cause of the symptoms after oral exposure is probably consistent of silent, subclinical phases and many other mechanisms that have yet to be discovered. However, the spreading of the bacteria has been discovered. Once in the cytosol, the pathogenic strains will polymerize actin filaments and act as the driving force to be able to move around the host cell until it gets a chance to be under the surface of the host cell. Once the bacteria has made this contact to the surface of the host cell, it causes an extrusion to form. The extrusion can penetrate the neighboring host cell. The second host cell can engulf these ramifications containing the living bacteria. This mechanism of cell-to-cell spreading avoids the host cell defense mechanism. Here's a picture of, on the right side of this picture, we have the method of contamination of the foods that we consume. It enters in through the esophagus, enters into the stomach, which it can survive the varying pHs and temperatures, and once it goes through the intestines, it is then absorbed, and from there goes through translocation using the actin filaments, and from there our immune response could cause one of those symptoms to occur. Diagnosis and treatment. Antibiotics. The doctor would typically run tests to rule out salmonella, botulism, and E. coli first. However, if these come back negative, the doctor would request a lab to culture the patient's blood, cerebrospinal fluid, or amniotic fluid on a medium selective for Listeria, like rapid L agar. The common antibiotic is penicillin or ampicillin. If the patient is allergic to penicillin, trimethoprim, sulfamethoxazole is used. Antibiotics are given promptly to the pregnant woman so that they can prevent the infection from infecting the fetus or newborn. Babies with listeriosis are given the same antibiotics. Prevention of dehydration, because there's a lot of loss of fluids through the symptoms. And a change to a mild food diet consists of avoiding spicy food, fruit, alcohol, caffeine, from oily foods, and milk. According to the CDC, there are a few recommendations to avoid the infection with Listeria. Raw food should be handled with caution. Wash knives, cutting boards, gloves that have been used when handling raw foods. During a time of most susceptibility, avoid hot dogs and deli meats until reheated to the point of steaming hot. Stay away from cheeses such as feta, brie, or Mexican-style cheeses. Refrigerated smoked seafood should be consumed with caution. Many different companies have developed a bacteriophage treatment. A bacteriophage called Listeriophage P100 has been proposed to be an additive to control the bacteria. The FDA just recently approved of a cocktail of 6 bacteriophages from Intralytics and another one from EBI Food Safety designed to kill Listeria monocytogenes. This spray would be used on fruits and ready-to-eat meats in the future. This is my work cited, and thank you.